from Yoshinari Yoshida, who is familiar to lots of us, but if you come into our seminars, of course, is familiar as in his role as seminar organizer, but he's going to step to the outside of the table room today to give us a talk, which is wonderful. Yoshi is a postdoctoral fellow here at Egenis, has been since October 2023, right? Yeah. And before that, he studied at the University of Minnesota and got his PhD in May of that year, 2023, with Alan Love, supervisor. And before that was at the University of Kyoto. And Yoshi's interested, I suppose, in models and generalization in biology in general, but particularly developmental biology, and with, I think it's fair to say, a sort of philosophy of science in practice type orientation approach to those topics. So we're really delighted to have Yoshi's talk today. We'll follow the usual structure of Yoshi's talk of around 45 minutes or so, then we'll have a quick comfort break and then come back for questions and begin with any questions from any career researchers if there are some. And so I think what remains to, I'm just looking at these lovely pictures here, so what remains to do. So Yoshi's going to talk to us about comparing multiple inaccessible systems through biological modeling in the case of cellular anthropology.
Thank you, Adam. And thank you all for coming. I'm very glad to have a chance to this opportunity to share my research with the Egenis community. So yeah, as the title suggests, I'm going to talk about like modeling in a specific context, like combination between in vitro technologies and comparative biology. So first, what are in vitro models? In vitro models are like cells or tissues maintained and cultured outside of your body and studied as models of actual organs or living organisms. So you can think of, yeah, like cells cultured on a dish as model of you or human or, yeah, things like that. And philosophical discussions about in vitro modeling have focused on how they serve as models of human development, physiology, and pathology. Basically, like focus on the narrowly on the biomedical context. And here I have very simplified schematic representation. You have in vitro models, which represents the target system, human body. And this is like fine. It makes good sense that many philosophers are interested in the use of in vitro models in this type of like biomedical context, because that's like what many people care about. But the scope of application of in vitro modeling is much broader and like expanding, especially recently. And this presentation focuses on how in vitro models serve as models in comparative biology context. So the very schematic basic idea is unlike this type of situation, in comparative biology, in vitro models, you can think like these are in vitro model 1, in vitro model 2. In vitro models are used to represent different organisms, basically different species, and it is motivated by the interest in similarities and differences between those species. And the basic important assumption is those organisms are both or all experimentally inaccessible in a certain sense. So researchers make those in vitro models to represent those organisms, and by comparing those in vitro models, they try to learn about similarities and differences between the target systems. That's the basic structure. And here are main points of my presentation today. As an example of in vitro modeling in comparative biology, I discuss a recently emerging area called cellular anthropology. I will explain shortly what this area is. This area, cellular anthropology, compares in vitro models derived from different ape species to generate knowledge about similarities and differences between the human and other apes or I would say like the similarities and differences among different ape species, but especially between human and other apes. Yeah. And cellular anthropology raises several interesting, I think, philosophical issues. And I'll briefly mention a few of them, but one of them concerns experimental control. So the latter half of this presentation will focus on this specific issue, experimental control. There, I will argue that although experimental control is typically understood as fixing the background constant, the case of cellular anthropology suggests that scientists sometimes treat two systems differently to achieve experimental control. Yeah. So if this were like regular conference presentation, this last point would be the thesis. But I made the presentation a bit broader, in part because I have more time, obviously, and also Egenis community includes this last point is basically philosophy of science type conclusion, but because Egenis community includes not only philosophers but also social scientists, anthropologists, and this earlier part of this presentation will be made somewhat broader and kind of open-ended, and also at the end of section 3, I will mention some ongoing and future investigations in my project, so that I can receive feedback from broader or different perspectives, not only specifically about this philosophical topic. Yeah. So I should have explained the outline first, but I've done the brief introduction. In the next section, I will briefly discuss in vitro models in clinically oriented, I would say mainstream stem cell biology, basically in the biomedical context as a contrast case. Yeah. And in section 3, I introduce cellular anthropology, what it is, what motivates it, what researchers do in this area. And then in section 4, I'll have a bit more focused discussion about experimental control in cellular anthropology, but in vitro comparative biology more generally. I think this area, in vitro comparative biology, including cellular anthropology, raises an interesting philosophical issue concerning experimental control. Okay, so let's move on. In vitro models in clinically oriented stem cell biology first. So just to give you background information, there are many different types of stem cell, many different types of in vitro models, but I'm gonna focus on pluripotent stem cell lines. Pluripotent stem cells are those cells that can generate themselves, pluripotent stem cells can generate more pluripotent stem cells and also generate more differentiated cells, which eventually become various cell types. So basically, pluripotent stem cells can produce all cell types that constitute an organism. Yeah, neuronal cells, skin cells, kidney cells, blah, blah, blah. There are two important classes of pluripotent stem cell lines. One is embryonic stem cells. Embryonic stem cells are derived from early embryos of that organism. Yeah, and just like early embryos can eventually produce wide range of cell types, those embryonic stem cells have the capacity to generate many different cell types. The other class of pluripotent stem cells is induced pluripotent stem cells, or iPS cells. Unlike embryonic stem cells, iPS cells are derived from somatic cells, which are adult, already differentiated, mature cells that constitute your body. So for example, researchers can collect tiny skin samples of yours and introduce certain transcription factors and culture the cells in like specific conditions and make those skin cells, originally skin cells, reprogram and like basically go back into a stem cell-like stage. Yeah, so iPS cells exhibit similar differentiation and proliferation capacity to embryonic stem cells. So they also can produce a wide range of cell types. Yeah. And these two classes of pluripotent stem cells are widely used as model in biomedical research today. So they serve as models in a number of different ways. They can be studied as models of early embryogenesis, for example. Or, as I said, these cells, if put under certain like appropriate conditions, they can differentiate or produce more differentiated cells or more specified, specialized cells. So by like producing those different types of cells, researchers can also study the formation of certain tissues or organs by using those derivatives of pluripotent stem cells as models. Yeah, and here you can see the word organized. Organized are, you can think of organized as like a specific type of in vitro models that are generated from those stem cell lines. And organized are like very simply put, three-dimensionally structured three-dimensionally structured systems that are supposed to recapitulate specific organs or tissues. So there are different types of organized, like brain organized, skin organized, kidney organized, blah, blah, blah. Yeah. So you can think of organized as a like way of studying specific tissue types, organ types, by using those pluripotent stem cell lines. Okay, so this is a background. Now, yeah, a bit more about how those stem cell lines work as models. Basically, like, of course, the most important thing is that they represent early embryonic development and formation of various tissues. Yeah, some of which are experimentally inaccessible. For example, like early embryogenesis of humans, it's not easy to, well, basically impossible to study directly. So in that sense, early embryogenesis of humans is inaccessible. And those pluripotent stem cell lines can serve as models of those processes. Yeah. It is also important that, so like whenever you think about or whenever you assess the modeling representational capacity of a certain biological system, it is important that it is important for you to consider not only resemblance and differences between the model and the target system, but also like many properties or factors that make them tractable or convenient models. And these include like material features of the models. For example, like small size makes it convenient because you can keep many in your laboratory. Large offspring number, rapid growth rate. These enable like you to conduct experiments many times, many times, things like that. Robust processes of reproduction and development, tractability, simplicity, and accessibility to observation and measurement, things like that. Ethical considerations are also important, of course. So, for example, one strong motivation for many researchers to use in vitro models is to replace animal models. It's commonly assumed that it is ethically better to use in vitro models than to sacrifice many organisms as models. Yeah, and there are like political factors, social dimensions. There are like, yeah, many factors are influencing scientists assessment and evaluation of how well stem cell lines can serve as good models. But yeah, like just pointing out these, there are many different factors will suffice here. And Fagan, the philosopher of stem cell biology, Melinda Fagan, points out that there are many different stem cell lines, and they work together to represent a specific target, which is early human cell development. So I don't have enough time to explain this diagram in detail, but the basic idea is before embryonic stem cells were established for humans and mice, cancer-derived stem cells were used as models of embryonic development. And but in 1981, mouse embryonic stem cells were established, and then 1998, I think, human embryonic stem cells were established. And then iPS cells came in 2006 and 2007. So like, and in this process, researchers have generated new stem cell lines by importing protocols from existing stem cell cultures, and also existing stem cell lines were used as like contrast. How to say, like newly established stem cell lines were evaluated and characterized in comparison with existing already established stem cell lines, things like that. Yeah, so Fagan argues that stem cell models are not replaced by subsequently constructed stem cell models. Rather, each continues to be used. The result is an increasingly intricate tapestry of models interrelated to form a complex representational fabric. This suggests that there is some overall target of interest not adequately represented by any of these models alone. That target, I suggest, is not broad but complex early human cell development. This complex phenomenon is represented by an ever-expanding family of related models, each narrowly targeting a different aspect of this complex phenomenon of interest. So, yeah, I think that quote explains clearly enough, but there are many such different stem cell models, and in this like clinically oriented stem cell biology, those different stem cell lines work together to represent this specific target of interest, early human cell development. That's the picture Fagan is presenting here. And I find this picture convincing in the context of, again, like biomedical, clinically oriented stem cell biology. But my point of this presentation is basically I'm looking at a different area where stem cell models are serving a bit different function compared to those mainstream stem cell biology. So let me talk about cellular anthropology now. To introduce what cellular anthropology is, I first have to explain what apes are. So this is a family tree of certain primate species. Apes are a group of tailless primate species, including ourselves, chimpanzees, bonobos, gorillas, orangutans, gibbons, yeah, those primates. Yeah. Apes are different from monkeys. Monkeys are more distantly related to us, like this. And there are two major groups of monkeys, so-called old world monkeys and new world monkeys. And monkeys include macaques, drills, baboons, marmosets, etc. And so apes and monkeys are like distinct in terms of like phylogenetically speaking different, but for the purpose of this presentation and also in the context of stem cell research, a crucial difference between apes and monkeys is that invasive experimental research is not conducted for apes, but conducted in monkeys. For example, certain monkey species like crab-eating macaques or marmosets are still widely used in cognitive sciences, molecular biology, also biomedical context. They are used in invasive experimental research, which is not the case for apes. And apes are experimentally inaccessible for ethical and practical reasons. They are all endangered species, not easily accessible. Their body sizes are basically generally large. They have higher cognitive capabilities, which raises ethical concerns, and their reproduction time is relatively long, etc. Only chimpanzees used to be an experimental organism in biomedical research, for example, like HIV research in many countries, like a few decades ago. But since around early 2000, in many countries, the trend has been increasing to stop using chimpanzees in biomedical research. And in 2015, NIH announced they would no longer support biomedical research on chimpanzees. And as far as I know, since then, in all countries, chimpanzees are no longer used for biomedical experimentation. Yeah. So both ethically, practically, apes are not accessible targets. But at the same time, there is a good reason for why many researchers would be interested in studying apes, because they are so close to us, of course, obviously, and many human-specific traits that interest biologists and anthropologists as well have evolved through and after divergence of ape species. Many people would first think about expanded cortex, or larger brains, or upright walking, another characteristic feature of humans, naked skin, white eyes, relatively flat facial morphology. There are many such features that people think as human-specific that have evolved relatively recently. And because those traits have evolved relatively recently, if you want to study evolutionary origins of those traits, comparing our development and physiology with those of our closest relatives would be really important or informative. Yeah. But of course, those organisms themselves are not accessible. To overcome this situation, recently some researchers in different parts of the world have applied stem cell technologies to study apes. And these are just selected examples, and I can show many more articles. And those researchers often use words like terms like cellular anthropology or cellular primatology to refer to what they do. So in this quote, the authors say, "Our work provides a wealth of candidates for future evolutionary studies and demonstrates the value of cellular anthropology, a strategy of using in vitro derived embryonic cell types to elucidate both fundamental and evolving mechanisms underlying morphological variation in higher primates." Or in this another article, the authors say, "Cellular primatology, which is comparative studies focused on the cellular process underpinning the phenotypic variation among humans and non-human primates, including cell development, or molecular features, tissue composition, and organization." Yeah. So in this presentation, I'll use cellular anthropology, basically in this sense, the like use of in vitro models to study different primate species, especially apes, in the comparative context. And what do they do? What do cellular anthropologists do? I've already shown this figure to explain iPS cells and embryonic stem cells. So in the context of cellular anthropology, embryonic stem cells are not available because invasive experimentation cannot be conducted. So it relies on iPS cells. So researchers can collect tissue samples from captive apes, like skin samples, blood samples, or urine samples, and again, like introducing certain transcription factors and culture those cells under certain conditions to reprogram those cells to become stem cells. And they may study those iPS cells directly to learn about early embryogenesis in those ape species, or they may artificially differentiate those iPS cells into specific cell types, or like form organoids to learn about tissue formation or organ formation in specific species. Yeah. So a bit more concrete example. I don't have, again, time to explain in detail, but in this article titled "An Early Cell-Shaped Transition Drives Evolutionary Expansion of the Human Forebrain," the authors generated brain organoids of human, gorilla, and a chimpanzee. And brain development goes through multiple stages. In early stages, your brain consists of not fully mature cells called neuroectherial cells. Neuroectherial cells proliferate actively, and at this stage, brain expands laterally. But in the next step, neuroectherial cells start producing a bit more differentiated cells, which eventually become neurons. And in this later stage, brain starts expanding radially. Yeah, so this is a stage of producing more neurons. So early expansion and then producing neurons. Very simply put, yeah. In this study, the authors discovered that this early stage of neuroectherial cells expansion or proliferation and lateral expansion stage is prolonged in human brain organoid compared to gorilla and chimpanzee brain organoid. And this seems to suggest, this difference in temporal regulation across species seems to suggest that this prolonged expansion stage contributes to our expanded brain, evolutionarily speaking. Yeah, they don't claim that this is the cause of expansion of our brain, but one contributing factor. Yeah, so this is an example. And again, we can understand what is going on with this very simplified picture. So they want to know about what are similar and different between humans and other apes. To investigate this, they created human iPS-derived models, brain organoids, and chimpanzee brain organoids, gorilla brain organoids. They compared those in vitro models, found some difference, namely this difference in temporal regulation of brain development, and that suggests a difference between the target systems. Yeah, that's the basic structure of the reasoning, I would say. Yeah, and this is a bit different from the picture Fagan presents, because Fagan's point was like many different stem cell lines are used as models together to investigate a specific single target system, namely early human cell development. Yeah. Okay. So this is the end of the first half of the talk, and I must say there are interesting philosophical issues in cellular anthropology, and I mentioned just a few of them here. So, for example, how do stem cell researchers collaborate with primatologists and zoos? Because to conduct this type of research, at least stem cell researchers have to acquire cell samples of specific ape species. And practically speaking, the only available source is zoos or maybe sanctuaries. And so they have to interact with zookeepers or veterinarians, or sometimes primatologists, physical anthropologists. But of course, researchers in those areas usually don't share communities. So how do they collaborate? And I conducted some preliminary interviews in Japan and in the UK. And I found that in Japan, an information platform for primatologists. So not the platform is for the primatologist community, and not about like stem cells or anything. But such a platform for primatologists plays an important role. Basically, it mediates interactions between stem cell researchers and zoos or veterinarians. Yeah. Whereas, at least in a lab in the UK, I have visited interactions between stem cell researchers and zoos, or dependent on personal and individual interactions. Yeah, I have several slides to explain this case in Japan. And if you are interested, please ask in the Q&A, and I'm happy to explain using the slides. But yeah, more generally, how do different cultures of stem cell biology, primatology, physical anthropology, and zoos shape cellular anthropology in different countries? I'm very interested. Especially like Japan, my home country, has unique traditions both about stem cell research and primatology. In stem cell biology, like Japanese stem cell biology in Japan is aimed very strongly at regenerative medicine, like very strong political goal. And in interviews, I've heard some episodes about like how those stem cell researchers working on apes experience interactions with those other stem cell biologists who are working hard to achieve regenerative medicine. Yeah. At the same time, Japanese primatology has a very another strong tradition, kind of like sociological tradition of studying primates, and I'm interested in how they shape cellular anthropology in Japan, and of course both stem cell research and anthropology, primatology, and zoos have different features in different countries. So I'm interested in this type of question, but it's ongoing, still very preliminary, and but I really appreciate any feedback or questions about these features. Another more kind of theoretical, philosophical question is how can and should scientists integrate model system strategies common in stem cell biology and comparative strategies common in evolutionary biology to inform human evolution research? And the remaining of this presentation focuses on a specific issue related to this question, namely experimental control in vitro comparative biology. I'm aiming at 15 minutes talk. I'll do my best. I still have 15 minutes. Okay. Yeah, so experimental control. I think you all know very well that experimental control is a widely shared ideal in science, like contemporary science. In a recently published booklet, Shikori says that she talks about the comparative experimental strategy or design in which a target system or experimental group receiving treatment is compared with a reference target system or group that does not receive treatment. So a super simplified toy example is something like this. You are interested in the influence of watering on plants. And so in one group, you water. In another group, you don't water. And you observe what happens, like what differences result. And Shikori says the background, it is often said, must be held fixed. So in this toy example, you must be careful to fix everything else the same. Like only difference is watering or not watering, and soil, light, atmosphere, those conditions must be the same in order for you to attribute any differences you discover between the two groups to the treatment, watering or not watering. Yeah, a simple idea. Everybody, all of you know, right? Yeah, so that the effect of an experimental intervention can become manifest to the experimenter. And I would say like control is typically understood as putting the systems or groups in the same material condition, except for an intervention like this. Yeah. Now, in cellular anthropology, too, control is pursued. Researchers try to use the same protocol so that any differences in cultured cells are attributed to the species difference. So you can see them like analogously. They make human iPS cells and chimpanzee iPS cells. They try to use the same culture and differentiation protocols so that any differences discovered between these models must be attributed to the species difference. Yeah. Now, there are different protocols for iPS cell generation, as well as formation of organoids for a particular tissue type. So this is just an example, and even this is a like very simplified picture. So like from embryonic or iPS, embryonic stem cells or iPS cells, there are like different steps to take to generate brain organoids, and each arrow in this diagram involves like many procedures. So yeah, what I want to say is just like there are variations of protocols. Even you focus on protocols for generating a specific organoid type, like brain organoid. There are multiple protocols. So yeah, it makes sense that like researchers want to use the same culture and the differentiation protocol when they want to compare two systems. Because if you use different protocols and find some differences between the systems, you are not sure if the difference is coming from the difference in protocol or difference in species. So for example, in that like brain organoid research I briefly introduced, when talking about their experimental procedures, the authors say we optimized the protocol so that an identical method could be used for all species, resulting in highly comparable telencephalic tissues. This was vital to be able to attribute any differences to species-specific divergence and not protocol variations. So you can see like the authors are very aware of this like importance of control. And I'll explain the latter half of the quote soon. But before that, some researchers working in this area even emphasize this as a strategy to overcome species differences. So this is not a case in cellular anthropology, but it is still a case of in vitro comparative biology. This group of researchers generated in vitro models derived from mouse, rabbit, cattle, rhinoceros, human, and marmosets. And what they wanted to do was basically they are interested in how developmental tempos are regulated differently in different mammalian species. And as is obvious to you, these mammalian species have radically different body sizes, very different lifespan, so different, yeah. But they want to learn something about something in common, what are different, yeah. So this is a different paper, but the same author, yeah. They say direct interspecies comparisons of developmental tempo have remained challenging due to the different growing temperatures and uterine environments of individual animals. In vitro differentiation of pluripotent stem cells can be used to recapitulate cell or tissue autonomous developmental processes and study them under similar experimental conditions. So even though in actual development, mouse embryo and rhinoceros embryo are they grow in very different physiological conditions, by using in vitro technology, they can compare embryonic cells of those species under similar conditions. That's one strength of this approach, they say. Yeah. So I have emphasized the importance of control in vitro comparative biology. But it is not always possible to use the same protocol for interspecies comparisons. This is because a protocol that works for a cell line, individual, or species might not work for another. It is known that there significant variability in whether a specific protocol works across species. Yeah. So one iPS cell generation protocol that works for human cells might not work for gibbon cells, say. Yeah. So practically it sometimes doesn't work. Even beyond that, there are sometimes good reasons to use different protocols to generate comparable models. And a suggestive quote is, the latter half of this passage I have shown before. So as I said, they optimized the protocol so that the identical method could be used for human, gorilla, and chimpanzee cells. But they noticed that prior to neural induction, chimpanzee embryonic bodies are just early stage of organoid formation. Yeah. Chimpanzee embryonic bodies progressed faster than human and gorilla embryonic bodies, potentially reflecting the shorter gestational period of chimpanzees. However, the difference in timing may also be a result of cell line differences and further studies needed. Nonetheless, in order to obtain comparable tissues of the proper identity, timing was optimized and chimpanzee organoids were transferred to neural induction 2 days earlier. So in this video, they explain this. So this is steps or processes to generate brain organoids. And in chimpanzee cells, they use shorter culturing period because they judge that would be appropriate to generate comparable models. And this might reflect the shorter gestational period of chimpanzees, but yeah, as they say, it requires further investigations. But anyway, yeah, this is what they did. Now, this specific case is just it's basically practical, coming from the practical limitation, but it also suggests an interesting idea about experimental control, I think. which is there are different ways of implementing control to generate comparable models. One is to use the same protocol. This is the like widely shared idea. Everybody thinks like control experiments, yeah, like use the same material setup. That's the shared idea. But another way of implementing control is to adjust protocols to reflect differences in the natural developmental processes between species. And I emphasize that this is not abandoning control. This is a way of implementing control in the way that reflects differences in the target system. Yeah. I want to also emphasize that these are not merely different strategies concerning what factors to control. These are also different interpretations of what it means to treat cells of different species in the same way to make them comparable. Let me say a bit more about this. Yeah. So the first option, treat cells in the same way, means how do you say? Sorry. Yeah. So I said these two ways are associated with different interpretations of what it means to treat cells of different species in the same way. And in the first option, treating cells of different species in the same way means put them in materially the same condition. Yeah. And I think I don't need to explain anymore. Yeah, because this is basically, this accords with the like generally accepted idea of experimental control. But in the second option, treating cells in the same way means adjusting protocols to put the cells in corresponding developmental states. Yeah. And in another article coming from the same laboratory as the authors, the same laboratory who published this study says, so in this paper, they are not comparing different ape stem cells, but they are developing a protocol that works both humans and mice. And they are explaining that given this protocol seems to recapitulate early cerebellar development in human organoids, we next explored whether this approach could also be applied to mouse embryonic stem cells and enable cross-species comparisons of cerebellar development. To achieve this, we made brain organoids using mouse E14 embryonic stem cells. Notably, brain development in mouse occurs at a much faster pace compared to humans. Therefore, protocol steps were scaled according to mouse developmental tempos as previously performed. So I think here you can see like even more clearly these the idea behind the second option. Yeah. To generate comparable model, they decided to use different protocols. And sorry, I think it's hard to read, but this is basically this diagram shows different paces and stages of brain development across human, macaque, and mouse. Yeah, and you can see so blue is humans, red is macaques, yellow is mouse, and you can see different features of the brain, or different behavioral features appear at different points of development, right? And sorry, I forgot to include the bar that indicates the times, but anyways, the point here is that when you are comparing well-established experimental organisms, often this type of information is available because you have detailed descriptions of how that organism develops and how specific brain features develop at what point of development. And so you can have a better resource to decide which stage of your in vitro model you have to compare. This type of information is harder to achieve for organisms like apes, precisely because they are not experimentally accessible. So yeah. Anyways, so let me say a bit more about the two ways of implementing experimental control. I would say the two ways are associated with different methodological orientations. The first option, using the same protocol, basically compares in vitro models of different species in conditions that are as similar as possible. This will reveal cell or tissue intrinsic or autonomous differences across species. We can think of this as a kind of like methodological reduction to the cellular or tissue level. Whether the identified differences are actually relevant to phenotypic variation in vivo must be confirmed by using other experimental methods such as xenotransplantation, species hybridization, or interspecies chimerism. Yeah. In contrast, the second option, adjusting culture protocols according to differences in the natural developmental processes across species, this will pursue protocols adjusted to different species. And doing this requires knowledge about in vivo development of the donor organism, which is not easily available, as I've said, for non-experimental organisms like apes. So it's easier to establish such adjusted protocols for organisms like mice, zebrafish, harder for organisms like apes. There are also practical limitations, or like researchers would have to think where to stop, because there are so many factors that they could potentially adjust. Like I have talked about temporal features because that's the like major factor, and probably temporal factor is relatively easier to handle because for example, like gestation period, even if the organism of interest is not experimentally accessible, you can at least know gestation period relatively well, for example. But there are things like, yeah, body temperatures or like hormonal physiological environments in uterines. There are many things that would be different across species, and if researchers pursue this way, they have to think, yeah, where to stop. To what extent they try to adjust protocols, what factors they consider. Okay, and I'm not gonna say like anything about which approach is better or yeah, because I think these are both legitimate approaches. They just reveal different types of informations. For example, like using the same protocol, it is a legitimate way to generate knowledge about cell or tissue intrinsic differences. Just to establish the relevance of the discovered differences to like actual in vivo or phenotypic differences, they have to do certain like additional experimentation. And then the same applies to the second approach, like it is legitimate way, but it has its own advantages and practical limitations. So finally, I'm gonna briefly talk about if this issue is unique to in vitro comparative biology. I don't necessarily think like this is absolutely unique to in vitro comparative biology, but maybe the issue is kind of salient in vitro comparative biology because the following features are combined in vitro comparative biology. One is it compares different things through intricate experimentation, and the other is in vitro models are highly situated models. The first point, it compares different things. Probably some of you have like thought about this when I presented these figures. Typical control experiments compare things of the same kind like the same plant, for example, same plant species, with or without a treatment. Whereas in cellular anthropology or in vitro comparative biology more generally, it compares different kinds of things, for example, human, chimpanzee, and gorillas, through intricate experimentation. This is one feature that is important. But this itself is not special. Like there are fields of science that study that conduct cross-species comparisons using certain experimental setup. I want to say this feature is combined with the other feature, and that makes in vitro comparative biology a bit unique. And this feature is in vitro models are highly situated. By this, I mean in vitro models can represent the targets only because they are put in very specific experimental circumstances. In this sense, they are highly situated in the sense, Anthony et al. 2014, the concept of situated models is basically like simply put, like experimental conditions surrounding the organism or the biological system is a crucial part of their modeling capability. Yeah. So, yeah, for example, like originally the cells are just like say skin cells. If you do nothing, they are just skin cells, and like they would never represent brain development, right? Those collected cell samples can represent brain development precisely because they are put in very specific, complex experimental conditions. Yeah, so in this sense, cultural conditions are a crucial part of their modeling capability. And not only that, in comparative biology, in vitro models often must be co-situated to enable precise interspecies comparison. Yeah, because like using different protocols can produce different in vitro models. Yeah, so in vitro comparative biology, these two features are combined, and that raises this question of what it means to treat multiple in vitro models in the same way. Like, yeah, does it mean to provide the same material conditions or use adjusted protocol? So I don't know, this is an open question, but when these conditions are both met, the similar issue of how to treat in the same way would arise in other areas, but I'm not sure about other areas. Okay, so to summarize this section, control experiments are typically understood as putting systems or groups to be compared in the same material conditions so that any differences observed can be attributed to the treatment of interest. But this is not necessarily the case. There are cases in which scientists use different experimental conditions to control experiments. And this also suggests that experimenters make decisions not only on what factors to control and how, they sometimes have to decide what treat in the same way means. And different interpretations of this question are associated with different methodological orientations and hence like having different epistemic implications. Okay, so conclusion is just like repeating main points. Cellular anthropology compares in vitro models derived from different ape species to generate knowledge about similarities and differences between them. This area and in vitro comparative biology more generally suggests that there are different ways of implementing experimental control to generate comparable models, namely using the same culture protocol or adjusting protocols according to differences in the organisms. And these are associated with different methodological orientations. Thank you for your attention.